Activation of the complement system occurs on surfaces, such as the surface of a bacterium, as shown here. In this example, the activation of complement is initiated by antibodies that bind to bacterial cell surface antigens. The complement component C1 binds to the aggregated antibody molecules on the surface of the bacterium. Binding of C1 to antibodies results in the activation of the constituent C1R protease, which in turn cleaves and activates the second constituent protease, C1s. Active C1s is able to cleave and activate the complement protein C4, releasing a small peptide fragment, C4a, which acts as an anaphylatoxin. The remaining large fragment, C4b, is an active protease and attaches to the surface of the bacterium. C4b now binds and cleaves C2, releasing a small peptide, C2a. The combination of C4b and the cleaved C2b creates a new active protease, C4b2b, also called C3 convertase. C4b2b binds C3, cleaving it to release another anaphylatoxin peptide, C3a, while the C3b fragment associates with C4b2b to create yet another protease, C4b2b3b, the C3-C5 convertase. Many molecules of C3 can be cleaved by the C3-C5 convertase, releasing many molecules of C3a. The resulting C3b fragments are able to covalently attach to the bacterial surface, decorating it with many molecules of C3b that can induce the phagocytosis of the bacterium. The C3-C5 convertase also cleaves and activates C5. Again, the small peptide fragment released by this cleavage, C5a, is a potent anaphylatoxin. The large fragment, C5b, acts to initiate the formation of the membrane attack complex, the terminal stages of the complement cascade. In this process, C5b assembles with C6 and C7. C5b itself is not membrane-associated, and the C7 molecule allows this complex to insert itself into the bacterial cell membrane. C8 also binds to the C5b, C6, C7 complex and inserts itself into the cell membrane. The C5b, 6, 7, 8 complex catalyzes the assembly of many molecules of C9 to create a cylindrical pore spanning the cell membrane and disrupting the ionic and osmotic balance across the membrane, thus killing the bacterial cell.